working as assistant professor in the Department of Biotechnology, welcomes you all in this beautiful session of Sunday's Talkie. And today we have a very special guest with us, Dr. Alpana Joshi. Before we move on to her session, I request our President, Professor Arvind Deshmukh sir, to kindly share the vision and mission of Microbiology Society India. Over to you, sir.
Nitika ma'am, I am in journey. I am traveling.
Okay, sir.
So
Okay, sir. So
I
I'll
will not
just
talk much.
Okay, sir.
Only I wish all the best for today's program. Thank you.
Okay, sir. Okay, sir. Go ahead with the session. And we have Dr. Alpana Joshi, research scientist, College of Agriculture and Life Sciences, Jeonbuk National University, South Korea. Her research interest revolves around molecular biology and genomics, functional analysis of genes and protein, phylogenetical studies, molecular marker development, and herbal medicines. She adds to her name many research publications in national as well as international journal of repute. Today, she'll be talking about a very important perspective. That is, she'll be taking us to a journey of microbiological and biotechnological aspects. So over to you, ma'am.
Thank you. Thank you, Nikita, for the nice introduction of my. Actually, I was trying to connect from my laptop so I can share my presentation, but I don't know why it is showing updates again. So do I need to show the presentation, or if I am talking, it is fine?
Ma'am, if you are facing any problem in showing the presentation, sharing the presentation, you can go ahead with the talk only. No issues. We'll be sharing it afterwards. We can share the presentation afterwards in the group.
I can share with you right now even.
Actually, ma'am, I'm also,
if you like.
Yeah.
Yeah.
I'm also facing the same
let me
problem.
See, yeah, if in the. Oh, okay. Okay, so
you can go
good morning, everybody.
Talk, yeah.
Okay, yeah, I am starting.
Okay,
so
ma'am.
Today I want to discuss about the DNA barcoding. Actually, I am currently working with the plants, medicinal plants, and the authentication of medicinal plants. So as we all know that medicinal plants are very important, because now we are more focusing on the herbal supplement rather than the chemical, because herbal is the holistic approach. And the main problem with the herbal product or the herbal extract is the identification of product, the authentication of product. So whenever, if I am a industrialist and I want some raw material of, suppose, tulsi leaf or some cinnamon bark, so it is so difficult to identify whether this plant is from that particular species or not. So the DNA barcoding is the molecular tool, and using that we can actually identify, characterize species or genus of any plant species, microbes, animals, like any species having DNA. Correct? Okay. So what is the DNA barcode? DNA barcode is the short sequence of DNA. The length is around 400 to 800 base pair, and these are the unique marker of any species, where the species showing the high mutation rate. Like during evolution, you will see these sequences are more as well as conserved as well as high mutation rate. If any changes in those conserved sequences, you will find the new species, the generation of development of new species. So these are just the DNA sequences, the marker sequences of any species, and this can be used as an important taxonomic tool. And this method is why this is more authentic, because of its reproducibility. They are very much accurate and rapid and high reproducibility. Currently, sound is coming.
I
You
request
think it's
everyone to please mute. Yes, ma'am, you can go ahead, please.
Yeah. So nowadays of course, we are doing DNA barcoding. You will find so many publications, so many people are doing DNA barcoding of different genes. But now the new thing is that we are doing molecular profiling as well as biochemical profiling. So DNA barcoding along with the phytochemical profiling like HPLC chromatographic or LCMS. Now, these are the method which can helps to identify the secondary metabolites present in any plant species. So the combination of both methods, molecular methods and the chromatographic methods is giving a advantage to check the quality and the authenticity of herbal products. Okay, so now like there are so many products. uh, I said aloe vera example, aloe vera barbadensis, whatever aloe vera we are getting, like we are using for the aloe vera gel or any cosmetic, or we are using the raw aloe vera, that is aloe vera barbadensis. So if you want to buy any raw material of aloe vera, aloe barbadensis species, and you that product can be adulterant as in Acacia, uh, oh, somebody can be who can host the meeting, because I think every time I have to admit the
Ma'am,
because
you go ahead with your
new
presentation. I'm adding. I am adding.
yeah, because new they are coming, so
Yeah.
I have to add every time.
No, no, no
Okay,
issues.
so
I'm doing that.
yeah, so acacia catechu, acacia is the thing like which is used. The acacia bark is used to extract the katha what we are using in pan. So the katha bark is the adulterant of acacia, aloe vera. Asparagus racemosus, Asparagus gonocladus. These are the both species of genus Asparagus. Asparagus racemosus are used to cure the stomach problem, ulcers, spasm, constipations. But the adulterant of the racemosus is gonocladus. So, like that, there are so many species. Example, tulsi, for tulsi, this one is genus species is Taniflorum. Taniflorum has a wide range of therapeutic potential. It has antimicrobial, antidepressant, antioxidant, anti-inflammatory, anti-diabetic, anti-carcinogenic. See, so many therapeutic uses. But the adulterant of the Taniflorum is Osimum basilicum. So with the DNA barcoding, we can differentiate, we can identify the true osimum using the barcoding at the species level. uh, so chandan, like the santalum album chandan, everybody is using chandan, but now you will find so many fake chandan. So if you want the authentic material of chandan, you are going to make some antimicrobial product using the chandan, as chandan is having the antimicrobial property along with the neem, uh, Azadirachta indica. So that time you have to check the product quality, authenticity, whether you are adding the right product or not. So this is also one of the scope of the molecular characterization. Okay, so in plants, like there are two type of DNA generally, nuclear DNA and the extranuclear DNA. Like DNA is present in mitochondria and chloroplast, and nuclear DNA. So what are the difference between two types of DNA? Generally, the chloroplast DNA is called most conservative DNA because there is no crossing over is there. Na? But in nuclear DNA, there is lot of crossing over, and during evolution, you will find a lot of changes because if we're talking about the eukaryotic DNA has a lot of junk material, but big introns, like introns are like large introns as compared to the exons. The non-coding regions are more. junk DNA, repetitive DNA, and so many other elements which is required for the DNA replication. So the junk DNA, the non-coding DNA, is the one which is mutated higher when the species get evolved. So during evolution, you will find a lot of changes in the nuclear DNA. So if you want to compare, if you want to do phylogenetic study based on the nuclear DNA, so you will face a problem because there is a lot of changes, rearrangement happens. So it is little difficult. It is little tricky job to get the correct phylogeny based on the nuclear sequences. But still, there are some universal, I can say, universal marker of some family, like identified locus, which we can use as a molecular marker for the fingerprinting study, for the barcoding study, like the here. Let me check if I can mm, share. Mhm. No. So, like the mic, ITS1, ITS2, internal space, transcribed spacer. This is the part of DNA, MATK, mature HK. So there are different marker: ATP, PSB, YCF, RBO. So these are the examples of few universal marker which we are using to identify the particular species. In microbes or in prokaryotes, mostly we are going for the 16S RNA sequencing, where we are identify their V3, V4 region, or V1, V2, V3, V4. So V2 and V4 and V3 or V4. So we can design the primer based on these regions and characterize any bacteria, any new bacteria, or any unidentified bacteria. Similarly, with the plant, these are the markers ITS, for example, ITS, which we can use to characterize the new species or unidentified species. So what is the application? So here actually we are talking about the application. Application is that we can assign, identify unidentified individual species. It helps in the discovery of new species, and this barcoding is useful to develop a genetic resources tags. We can identify any expressions, germplasm, as well as we can identify the cryptic changes, very any significant changes, very minute changes, but like significant changes which can change, which can make a new species. We can just can change a species completely. So that we can identify the cryptic changes. It also used in the DNA fingerprinting. Like DNA fingerprinting is used in the forensic studies, so where we are using the different markers, RFLP, RAPD we are using, and seeing their bending pattern in the forensic investigations. Okay, and to check the authenticity of raw material of medicinal plant which is sold in the market, at last, which we can use the barcoding in phylogenetics. So finally, we can search for the ancestry of, like, I will give you example of wheat what we are having currently, the cultivated wheat, Triticum aestivum. Triticum aestivum having three different type of genome, A, B, D, and now we know the parentage of Triticum aestivum. So there are different methods, like we can do GISH also, genomic in situ hybridization, fluorescent in situ hybridization. Okay. So similarly, the barcoding, the molecular characterization is giving us an idea or a documentation that they, these species might be the ancestor of our current, our cultivated hexapolyploid wheat, Triticum aestivum. So there are so many polyploid species are originated either naturally or using the plant breeding techniques. So with these molecular marker, we can actually study the diversity and also search for the ancestry. So the flow of the method is, if we have some identified plant, like how we can identify any plant species. First, by looking its morphological parameters, the flower, leaf, type. Okay, so like by visual marker, the morphological markers helps us to identify any particular species. But sometimes two species are very close and it is so difficult to identify the correct species. So what we can do, we can have a different samples and we need DNA. So any plant part can be used as the source of the genomic DNA. Mostly we should go for young growing fresh tissue, because after the genomic DNA isolation, we have to go for the PCR. So when we will have a genomic DNA, now the PCR. So for the PCR, we need primer, and this is the main tricky part. So the primer should be of specific type. So like like ITS, MATK, RBO, some family are very much explored. We know that for this family, if we use the chloroplast TRN primer or nuclear ITS primer, that will give good result. But some family, like currently I am working with the Rudraksha, Eganitrus. So I have some result also in the last slides. I will show you. So you will find this total 14 sequence of Eganitrus in NCBI, because very less work of sequencing is done on Rudraksha. Rudraksha. Um, so in that case, what we have to do, we have to look for the sequence of the genus Eliocarpus. It is Eliocarpus genitrus. We have to look for the genus Eliocarpus, and then should have a all sequence of Eliocarpus. So we can build a multiple sequence alignment, and then we need to look for the conservative sequence where we can design primer. Okay, so there are two things. If we know the species, then we can use the universal primer. If we don't know the it's unknown taxa completely, then we have to go for the related species sequence alignment for the primer designing. So after primer designing, we will have the PCR fragment of correct size, and then we have to go for the Sanger sequencing. So Sanger sequencing, like Sanger sequencing, I think everybody know it's very old method and common method. Sanger sequencing, with the Sanger sequencing, we can sequence like if you do both ends, like both ways, forward and reverse both ways. So around 800, maximum 800 base pair sequence. So as we told earlier, that barcodes can be 200, 400, 600. Any conservative sequence can be used as a barcode, as a marker of a particular species. So after single sequencing, we will have all the sequences, the actual sequences, and then that sequence then again need to go for the blast and another bioinformatic analysis, to check whether the quality of sequence is good or not. And then later, this sequence, like of course, we have to repeat the experiment few times with the different plant part, or I should say the plants of different places, different geographical region, the same species, to check whether this marker is unique or not. So the reproducibility is the first foremost criteria of the marker, because we are going to claim that this is a barcode. If it is a barcode of any species, means if this sequence is present in that particular species, so this is the X species. Okay, So this is the general methodology of the barcoding. Okay. So initially this thing was worked, this barcoding was developed in case of animal where cytochrome oxidase CO1, it was identified as the universal barcode in case of animals, because like whether you take mouse model or of course prokaryotes E. coli, it is completely sequenced. In plant Arabidopsis is sequenced. Now there's so many new plants are also sequenced, but these are the model plants. And in animals, in animal model, mostly we are working with the rats. Okay, so these are the barcodes was initially developed with the animals, and later it was found useful in identifying the new species, new taxa in case of plant. And what we are doing, we are using the same barcode to authenticate the herbal product which we are using for processing, not the live plant, any plant part, whether it is like any like powder of any like bark powder, because bark powder of, and if you have a bark powder of 10 different plant, it is so difficult to identify which one is chandan or any other plant. Chandan, of course, you will have some smell, but now you can add smell also, the fragrance also artificially. So, okay. So with the plant, so traditionally there are two different kind of barcode was using single locus and multiple locus. Single locus means the barcode of just one gene. We are using, suppose we are using X gene, one gene, and multiple locus simultaneously we are using two locus. Okay, let me clarify it. Single locus means X gene, so we will have the forward primer of X gene as well as reverse primer of X gene. So this is single locus. Multiple locus, X gene forward primer, Y gene reverse primer. Okay. So this it's called multi-locus barcode. So multi-locus barcode is more authentic because two different genes are involved. So okay, now then after that, this was the traditional word. After that, as the sequencing approaches are getting advanced, more cheaper, more affordable, so now we are doing the CP genome sequencing, chloroplast genome sequencing. So in the chloroplast genome sequencing, because chloroplast is a very small genome, easier to sequence, easier to assemble, easier to analyze, and like and after getting the assembled sequence of chloroplast, you can have a like multiple gene sequence that are conservative and can be used as a barcode, as a universal marker of a particular species. Super, it's called super barcode also. And then after that, specific barcode, the advanced new one is specific barcode, which is more accurate, more fast, because here we are using the specific primer. But the problem is with that specific barcoding, we should have the some basic genetic information of that is in species to develop the specific barcode. Otherwise, we have to go for the traditional method, single locus barcoding or multiple locus barcoding or super barcoding. If we can afford, we can go for the super barcoding because it will give you with the it will give us the complete sequence of the chloroplast. Okay, there are a few more terms like they are the approaches of the barcoding. Mini DNA barcoding. Okay, so mini DNA barcoding is the where it's like the as a term defined mini. Here we are using very small fragment, very small, less than 200 base pair. Amplicon length would be less than 200 base pair, and the primer sequence will be standard. For example, MATK, maturase K, or RBCL. These are the barcoding region which are generally using by different investigators, researchers to barcode the uh, different family, different species. Okay, and and the DNA barcoding integrated with the chromatographic fingerprinting. As we have just discussed that we can use the HPLC techniques or LCMS technique or GCMS techniques to get the idea of the fingerprint of phytochemicals present in plants, because some like secondary metabolites is the you will find in all plants. Each and every plant will have secondary metabolite, because these are the secondary metabolite production is the stress surviving strategy of a plant, again biotic or abiotic stress. So the general like alkaloids, phenols, flavonoids, all plant will have this kind of alkaloid, but some plant like if we talk about the turmeric or bitter gourd or capsicum, so all have a different like specific alkaloids, the marker compounds. Okay, so we can integrate it with the chromatographic fingerprinting with and the molecular techniques. Okay. So nowadays we are going for the next generation sequencing. The Illumina is the common platform everybody is using, because plant genome is bigger as compared to the microbes. And in Illumina we are getting kind of full coverage of all genome. So the next generation sequencing, unlike the chloroplast sequencing, have the more data, because the nuclear DNA is large and has more non-coding region, junk region, and that region can be used. Now this region can after sequencing. So this all the molecular data can be used to develop the RAPD markers, SSR markers, ISSR markers. SSR markers is the we are looking for the repeats reason. So if we are having all the uh, genome sequence, we can find the actual repeat reason present in the particular species, and then that information can be used to develop a marker of that species. And then we can study, we can like different type of molecular study. We can do genetic diversity study, we can do phylogenetic study, we can go for the phylogenetic. If somebody wants to alter or change the or modify the um, any genotype in response to the disease resistance, then also we can look for the changes, you know, because the plant breeders, what the plant breeders are doing, they are treating plants with different mutagen, mutation breeding, yeah, or the plant breeding in collaboration with the tissue culture, where we are producing the where we are exploiting the somaclonal variant. So the somaclonal variant also can be identified using the markers, the molecular markers. So the molecular markers applications are like so many in different fields. So the next generation sequencing gives a lot of data. And one more, the major marker we are using nowadays, SNPs. SNPs, like mostly in human disease analysis. Yeah, SNPs are very common in very reliable, very authentic marker, and mostly we are using in the human genetic diseases. But in plant also you will find so many paper. Like SNP is also very good marker to study the genetic diversity of any species. So these are the some approaches what we can use for the barcoding. Okay, so here I will tell you the success rate when we are using the single locus barcode. So there are four markers: ITS, ITS2, MATK, and RBCL. The genetic specificity of ITS, ITS2, MATK, and RBCL is more as compared to specificity. Means it is more easier to differentiate the species at genus level as compared to species level. Yeah, because same genus, example, you will find different kind of tulsi, lemon tulsi, shama tulsi, ram tulsi, and there are so many more tulsi. Now we will see like there are different, and it is actually some tulsi are very common. We can identify because it is our holy tree. Like other, but actually the species identification is not that easy, by just looking morphologically. So this locus can use to differentiate the genus, and there is, and similarly with the multi-locus barcode, we found that ITS METK combination, ITS2 plus RBCL, METK plus RBCL, tRNH plus PSBA. So these marker combination is also able to detect variation at a genus level, as compared to species level. Species level is also they are like around 90%. In METK RBCL it is 98%, 99%. But at genus level, these multi-locus barcode are giving 100% result. Okay. There is one more term is barcoding gap method. So barcoding gap method is like when we are doing barcoding, when we are having the sequence, it is not enough that you are doing just did PCR sequencing and now you have data, so you can do whatever you want. No, there are so many different like the bioinformatic tools, different methods, different criteria you have to follow whenever you are analyzing your data. Okay, so in barcoding gap method, uh, barcoding gap method differentiate the gap between the variation between interspecies and intraspecies. These gaps. So here we are identifying these gaps, the gap, the variation between interspecies or intraspecific. NJ tree method and neighbor joining tree, like maximum likelihood tree. So these are the approaches. And here we have found when we use the three different combination RBCL, METK, and ITS. So it gives good results. So it means like it is best when you are using the multiple locus in analyzing the data. Okay, so Iliocarpus genitrus is our sample. We are working on, our team is working on, and if you look for the nucleotide sequence of Iliocarpus genitrus, commonly known as Rudraksha, just 14 items are available, and not all the commonly used barcodes even not sequenced yet. So just 14, you can look for. So here the major problem was to identify the unique sequence, the conservative sequence which we can use to sequence our genitrus, to amplify our genitrus, because we didn't have a basic genetic information of genitrus. So we just isolated, we did in silico analysis. We have assembled all the uh, sequence of METK sequence of Iliocarpus, and then just run MSA by muscle program. You can use Mega, you can use Cluster, you can go for BioEdit. There are so many freely available programs are there which you can use for the MSA. And with that program, you can design primer. And also the NCBI portal also facilitate the primer designing. You can also use the Primer 3 Plus software. Okay, but when you have to go for the multiple alignment, so it is better to go for the maybe Mega or Biorit, because they are freely available, because you have to compare all the sequence, look for the variation, and find the better place where you can get the primer, and that primer, the potential primer, because you never know whether this primer are going to hit your gene or not, because the species you are going to work has no basic genetic information available of that sequence. So here, like you have to do hit and trial. So just go for try the different uh, like number of primer you can design. Okay, and also after designing primer, just when you are doing the PCR, so just try to modify, like just play with the thermal profile. Okay, because uh, we have a not we don't have the genetic sequence, so you are just looking for the sequence of the closely related species. So there must be some indels, you know, some SNPs must be there, and the variation must be there. So if you go for the less annealing, so there would be a more chances of uh, primer binding and the extension. So this is just the technical part. If somebody is working in that area, so maybe they can have a better understanding. Okay. Okay, so the MATK, MATK is the very known, commonly used universal barcode reason, and this MATK is the tRNA, transfer RNA. We all know the translation. In translation, the tRNA has a major role. So the tRNK is nothing. It is tRN, transfer RNA K is there. So 5 prime tRNK, 3 prime tRNK, and in between you will find the MATK gene, maturase K gene, and this gene is around 1500 base pair long. And if you have the known sequence, you can design primer of any part. Otherwise, just look for the universal primer. Okay, there is a database of the barcoding of life science database, BOLD, where you will find some databases of at least the known species or the commonly species. So this is also one of the good marker which we can use it. Okay, so what I did, I just took one sequence of MATK and just did blast. Okay, and after doing blast, I have a series of sequence, no, the blast results, series of sequence where I got the 100% hit. So I have selected all the sequences, mostly of the same genus. I have just omitted the because I found the hit in another genus also, but I have omitted the hit in the another genus. I have just selected the genus of Aleocarpus, and then I have just downloaded the complete FASTA file of all the sequences, the hit file, and then just open the mega. Okay, and build the alignment in the mega file. So here the I just look for the conservative domain where I have found the multiple matches. The most of the sequences are having some common motifs, some motif region, some common site, and that region was selected by me. So for the primer designing, and we just did like similarly design four pairs, four forward and a four reverse, total 8 primers. Okay, so here these are the some primer sequences which are studied by other people, and having the good results. So here I will share this PPT, and if you want, you can see it. This is taken from some paper, the primer sequences here, and then with that sequences, we just calculate the pairwise distance, genetic distance. Genetic distance is also the criteria when you are studying phylogenetic. When you are doing multiple alignment sequence, you should um, quantify the genetic distance to see how close they are. Which species, which genus can be the close relative of your plant or your sample, so you can like take the reference of that species or that genus. These are the just a very big files, so I just took the shot of mega. Here you will see all the Eleucarpus and then genetic distance. It's all the matrix. This is a matrix Okay. And then I have calculated the transition transversion rate, because when there is a mutation, like if one species is converting into the another species, so one species is changing. Yeah, slide is not visible because my system, my system is unable to um,
No, no,
log
ma'am.
in.
It's going
Yeah.
very well. It's going very well.
Yeah, no, somebody is saying slide. I know with the slide, it was more
No issues, ma'am. Ma'am,
sensible, but
I
because
already
without slide, I am just telling. I don't know whether it's
no
somebody is
issues,
getting it.
ma'am. We'll share your
Yeah, I
slide.
don't know why. What happened to like the Zoom? Why it is not working in my laptop? Again, it is asking for the update.
No
So
issues, ma'am. No issues.
okay.
Your presentation is very clear. Without PPT, also we are able to connect, and we'll be
Yeah,
sharing
if
your
somebody
slides
is
too.
working in that area, maybe they can connect.
Yes,
Otherwise,
yes,
I
ma'am.
know it would be little different. So
No,
okay,
no,
so
ma'am.
here like
It's
we have to
very well.
also look for the transition and transversion. Yeah, transition and transversion rate, because like during evolution, uh, like A is converted to T or T to C, any like transition and transversion, purine to purine or purine to pyrimidine. So here we found the nucleotide frequency, the A changed 25% and thymine uracil 25%. So all four, like they all have the equal frequency 25% in each when we have studied the maximum likelihood, like while we are studying the genus Eliocarpus. All the species available in NCBI in current date. So here we have studied around 86 nucleotide sequence of maturase K, MAT-K, and then I have just run the program, the three programs, just to see how they look if I'm running the like I'm on the doing the phylogenetic study. So not very clear data because so many, maybe there are though so many like overlapping sequences are there. So uh, here it is saying that some plants are outgroup, but I cannot find they are really outgroup, because here I can see like so many sequences are overlapping. uh, you know I think because these species are like religiously we like Rudraksha is very important in Hindu culture, but scientifically very less work has been done. But now like more people are doing medicinal work also, phytochemical characterization also people are doing, but this result is not very good. These things look very much overlapping, and also one of the reason is that people have submitted a small fragment of sequence, not like not the not the um, long DNA sequence are submitted, very small sequence are submitted, so not very good results are there. So but here you can see the tree. I will share the PPT after the presentation. Okay, one more very important reason is transfer RNA. uh, it's called TRNTLF. So TRNTLF, and this is these are the this reason is one scientist Taberlet, one paper, like this most cited paper I should say, because everybody is following that TRN reason, the primer used by Taberlet. And so here this reason is very much unique in each species, and this sequence you can use for developing a phylogeny of any species. So here also this is tRNA T region, L region, F region, and 5 primers are there, A, B, C, D, E, in different orientations. So you have to look for the sequence, orientation, how big amplicon you want. So and then you can have a good data for the analysis. And the ITS, similarly with the ITS, if you search for the ITS, you will have a ITS 1, 2, 3, 4 different primer combination for ITS. ITS2, I would say, is like more authentic. Like if you are studying the medicinal plant species, even the in the Chinese culture, they are using the traditional medicine, traditional Chinese medicine. So there are so many herbal plants, and like their herbal system should is more advanced. They are using more herbal product, and they have more scientific investigation as compared to us. We also have so many herbal products, but we are not using. We have not worked that much. It needs to be explored more in a scientific way. So this is the ITS reason. Here in ITS reason is it's a nuclear fragment where 18S rDNA. Then ITS1, this ITS1 is actually internal transcript spacer reason. Then 18S rDNA, 5.8 srDNA, 26 srDNA. So ITS1, ITS2. So these are the gap, and this is also very good reason which you can use to amplify your mm, sequence, and you can develop a really good authentic DNA barcode using this ITS reason. The another one is RBCL. So RBCL is a ribulose 1,5-bis phosphate carboxylase. This is large subunit reason, RBCL. So this is also the chloroplast marker, and it has a length of around 600 to 750 base pair. So this RBCL. The 5' region of RBCL is ATPB, and the other side is ACCD. So these two genes are also used as a barcode in many species. You can, like here you can go for the multilocus ATPB forward, RBCL reverse, or RBCL forward, ACCD reverse. In any any primer you can design based on the how long amplicon you want, because you have to look for the uh, like the amplicon also. If the amplicon is will be too long, then you will face problem in the amplification. Okay, so here you have to be careful. And the TRNH PSBA region, this two region also used in the multi-locus barcoding. So here uh, there is three conservative regions, C1, C2, and C3, which are located in the variable regions. C1 is a conservative, then V1 is a variable region, then C2 is a conservative, then V2. So it's like that, and intron exon kind of thing. So here the variation will be in the V1 and V2 region. So you can design primer with the C1, C2 region, and you will have a good barcode of any species. So in one slide, I am actually showing the primers what we have used for our study with the Ganidrus, and also I am showing the PCR gel of few markers here. Okay, and then in one slide, after the PCR, then we have just uh, here we have a sequence of one gene, just one gene. I have one gene sequence I have pasted forward and reverse, and also along with the chromatogram. So when you will send the sequence for the Sanger sequencing, you will have a two file, one file for the forward. If you have asked for the both end, so you will have two file, one file for the forward sequence, another file for the reverse. So what you have to do, you have to merge both sequences, and then you will have the complete gene, the sequence gene. And also you can check the quality of the sequence, the Sanger sequencing, with the chromatogram. And there is one software, sequencer, and sequencer is the paid version. You can I think Mega also added the sequencer file, so maybe you can use Mega. I haven't used Mega with the chromatogram, but this chromatogram, when you will see the chromatogram, you will see that each nucleotide has the different color of peak. So you can check whether these peak represent the nucleotide correctly or not. Okay, so like that you can check the quality of your sequencing, and the forward sequence you have to go for the reverse complement, and then you have to merge it to get the complete actual sequence. Okay, and there is one term after sequencing, the Fred quality score. Fred quality score, like here you will see the Fred quality score is 10, means 90% is accuracy, like one incorrect base in 10. So it is like that. Just the you should know the term, like 10, 1 in 10, 20, 1 in 100, or 50, 1 in 99.99%, almost 100% accuracy. This is the quality score of sequencing, because sometimes you will find the after Sanger sequencing you will have a like n. If n is represent the nucleotide, so because some sequencing problem, or if the DNA quality is not good, like after PCR you have to purify the that fragment. But if the fragment is not purified properly, or sometimes it is not stored properly and or get degraded, so you will not have the proper sequencing result. So that time the technical part is like you have to be careful, not very tricky. You have to be careful whenever you are sending your sequence. So otherwise you will not get the correct sequence. You will get the sequence, but it will be just only error, or the more background peaks, where like which you cannot use in your study. Okay, so this is the Fed quality scoring. Okay, so in the just I am almost done, I think. So in this last these slides, I have put my LCMS result, what I have found in case of the phytochemical profiling of the Genitrus. So I did LCMS and HPLC. So to get the complete fingerprint of the phytochemicals present in Genitrus. So now I have like maybe more authentic data of the molecular as well as phytochemical of Genitrus. So if I will have any uh, tissue or maybe new species of any other region of Genitrus, so maybe I can check, I can authenticate whether this is belongs to the Genitrus or not. So you can go for the GCMS analysis also, because GCMS also give the complete profiling, and even initially like initial analysis you can do with the FTIR, but FTIR like they are just with the basic method, which is giving you that yeah you have like something, but for the actual characterization, these are the fingerprinting method you should follow, or the otherwise like the if you are targeting the specific phytochemicals to just isolate the alkaloid, flavonoid, like that, and then just to characterize the individual alkaloid like the quercetins, gallic acid, so, or catechin. So that kind of compound you can actually identify with this fingerprinting method, and like that would be the unique thing of your sample, and can use that you can use as a marker. So this presentation was actually about the markers, but now the terms we are using the barcodes, like the barcode, but when we are doing shopping, when we are going mall, it has now each and every things has the barcode. If we scan the barcode, it will give the information that this is the brand name, product, price, like that. So if we scan the barcode, like with that, finally we can develop the barcode, and if we screen the barcode, um uh, so you will have all the information that this genus, species, this plant has that many, this is the geographical reason, that many variant markers. So this is the way of molecular characterization of any species. So if anybody has any questions, questions are welcome.
Thank you so
Thank you.
much, ma'am. It was a very well elaborated presentation, and without like sharing slides, we were able to get everything. I think my participants
Thank
and
you.
audience here fully agree with me at this particular point. And this was the first presentation I have ever seen. It was very clear, and it was like a connecting dot from that particular initial thing to the last one. Thank you so much.
Thank
And if
thank
anyone has any
you.
type
Thank
of queries, they can just come
Yeah,
forward.
if I can answer, then I would be happy.
Do we
If
have
somebody is
questions?
working in that area,
Yeah.
then it's fine.
So if anybody is working related to madam's area, he or she can very well connect to madam, and we'll be providing madam's email, and also we'll be sharing the PPT. So ma'am, I'll be adding you to the Microbiology Society International group, and you can just forward me the
Yeah,
presentation.
I will. Yeah, I will share even with you, no problem. The presentation. The presentation was just very normal presentation. It's not very. But yeah,
No
if
issues,
somebody
ma'am.
has any
Yeah.
technical information, if somebody is looking for working in that area, I
Hmm.
would be happy to help if I can.
Yes, yes, ma'am. Main
Okay.
purpose of conducting these lectures is like to connect is to connect with the people abroad and also the ones who are doing research in the related areas, so that we can put forward the
Yeah.
collaboration. So I would request
Actually,
everyone to reach out to
currently,
Madam.
yeah. Yeah,
Yes,
actually,
ma'am.
currently I'm working in the R identifying the R gene NLR part, but that work is in many initial stages. So I didn't present that part. Otherwise, I was planning to present that study, the how we can
Ma'am,
identify the R
ma'am,
genes.
we will be inviting you for the further lectures also when you feel that you need to interact with the students, faculty, and all. You can present your that idea also.
Yeah, sure,
So we
sure.
have yeah. So we have
Yeah,
this
yeah.
lecture
No,
series
so this is it will take
on Sunday.
time. I have just working in that area, so it
No
is not
issues,
done.
ma'am. No
It
issues.
really takes time. Okay.
No, no
Thank you
worries, ma'am. And it
everyone
was
for
wonderful
listening.
connecting with
I
you.
think.
Thank you so much. And
Thank
I'll be
you.
just sharing your email ID
Bye
with the
bye.
group. Bye, bye, ma'am. Take care. Bye, ma'am.
Yeah, sure. Okay, bye
Thank you,
bye. Thank
everyone.
you.
Take care. Bye, bye. See you
Bye.
on Sunday's Talkies. Thank you so much. Thank you.